In this video, we will discuss this urocanase limits cis-UCA facilitated immunosuppression. In the previous video, we have understood the skin microbiome limits this cis-UCA facilitated immunosuppressions. So, in this figure, researchers has also pointed out that this urocanase also limit this immunosuppressions, and it will facilitate bacterial growth that we have discussed in one of our previous videos. So how they check their hypothesis is, first they planned an experiment that is shown in this panel A, where they disinfect the mice, and after 1 day they treat the mice with either SFE, which has both HER2 and HER2 genes, or C. singularis, which does not have HER2 and HER2 genes. Then after 7 days they use the cis-UCA, and after 3 days after that they sensitize it with this 0.5% DNFB, and after 5 days they do a first challenge and then again sensitize it with 0.13% DNFB, and then check the ear swellings after 24 days, and also after 30 days they did their second challenge and check the ear swelling after 24 hours. This is 24 hours, not 24 days. And what they are able to find out is that in the case of control, where they do not use this as any of these bacteria, and they sensitize and challenge, they do not see immune suppressions. But when they use this, when they don't disinfect and then treat the mice with the cis-UCA, they see immune suppressions of around 60% and 83%. 83% in the case where they disinfect and don't use any bacteria. And this decrease for 83% is for the first challenge, and for the second challenge it is decreased, immune suppression is decreased by 78.3%. And thereafter what they did is they check it for this bacteria S. epithelium, its name is mentioned here, which is this S. epidermis and C. singularis. So for this S. epidermis, when they do not use cis-UCA, they see some level of ear swellings, but when they use S. epidermis with cis-UCA, and both of them are with disinfectant, they see a decrease of around 26.2%. And in the case for the second challenge, they see a decrease of around 28.5%. But when they use C. singularis, they see a decrease of around 73%. And in the case of this second challenge, they see a decrease of around 43%. So this indicates that this urocanase bacteria in which urocanase is present, which can be found out by this HER2 and HER2 genes are present in this bacteria or not, they will have a decreased immunosuppressions. However, for the case where they don't have any HER2 and HER2 genes, they see a high decrease in the immune suppressions. And this trend is visible in the second challenge also, where we can see that when they use S. epidermis, the decrease of the immune suppressions is around 28.5%, but for the case of C. singularis, the decrease is around 43.8%. and now they wanted to check whether it is because of urocanase or not. So they use the urocanase inhibitors, glycylglycine. So what they did is they use disinfect the mice and then treat it with this bacteria colonies and this glycylglycine, which is the urocanase inhibitors, and after 7 days they add this cis-UCA, and after 3 days after that they sensitize and check it for the first challenge after 5 days. After 24 hours they check the percentage of immune suppressions. Then after 30 days they do the second challenge, and after 24 hours they check the immune suppressions. So what they are able to find out is that cis-UCA induced immunosuppressions 24 hours after challenge. That is, in this case, they are able to see that when they use the inhibitors, the immune suppressions increases compared to the case where they do not use inhibitors. And for the cis singularities, they do not see any significant changes. Then for the second challenge, they see the similar trend, that is when they use for the S epidermis, when they use the inhibitors, urocanase inhibitors, they see increase in the immune suppressions. But for the cis singularities, they do not see changes. So this indicates that urocanase plays a role towards this immune suppressions, UCA mediated immune suppressions. If urocanase is inhibited, then it will not be able to suppress this immune response by this cis-UCA facilitated. This is what they wanted to point out in this figure, or the experimental results that are shown in this figure. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. This is the last figure of this paper, main figure. So in the previous figure, or in the previous results, we have seen that skin microbiome also affect this cis-UCA mediated immune suppressions, and in this we have understood that urocanase also affect the cis-UCA mediated facilitated immune suppressions. So in order to have the maximum immune response, or in order to modulate the immune response, the skin microbiome needs to be controlled, and at the same time this urocanase positive bacteria also needs to be limits down, so that we can fine tune the immune suppression that is needed in the patients to treat these skin infections or any skin diseases. That's all for this video. If you need to understand more about this experiments in detail, you can go to the reference that has been provided in the description. Thank you for your time. Congratulations.